Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine, best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about steroid hormones, steroidogenesis, its applications, and biosynthesis. This is our third lecture on steroidal hormones, in which we will discuss about adrenal cortex, gonads, and different pharmaceutical brands of steroidal hormones. Let's see aldosterone. This molecule consists of a total number of carbon atoms 21 and hydrogen atoms are 28 and oxygens are 5. There is a presence of carbonyl group at carbon 3 positions and one methyl groups at carbon 9 position, hydroxyl group at carbon 11 positions and an aldehyde group at carbon 13 position. In addition with the methoxy group which is attached to the carbonyl group at carbon 17 position. So the molar mass of this molecule is 360.450 gram per mole. Let's see the solubility, uses and sources of aldosterone. Aldosterone is produced in the cortex of adrenal gland and it is soluble in organic solvents such as ethanol, dimethyl sulfur oxide and dimethylfuramide, which should be purged with an inert gases. Aldosterone affects the body's ability to regulate blood pressure. It sends the signals to organs like the kidney and colon that can increase the amount of sodium the body sends into the bloodstream or the amount of potassium that is released in the urine. The next molecule is testosterone, which comprises of carbon 19, hydrogen 28 and oxygen 2. You can see over here in the structure of testosterone there is a presence of carbonyl group at carbon number 3 and a double bond at carbon number 4 in the first ring, in addition with the hydroxyl group at carbon 17 position. Testosterone is the primary male sex hormone and anabolic steroid. In male humans, testosterone plays a key role in the development of male reproductive tissues such as testes and prostate glands, as well as promoting secondary sexual characteristics such as increased muscles and bone mass and the growth of body hair. In addition, testosterone is involved in health and well-being and the prevention of osteoporosis. Insufficient levels of testosterone in men may lead to abnormalities including frailty and the bone loss. The chemical formula of testosterone is C19H28O2 and the molar mass is 288.431 gram per mole and melting point is 151.0 degree centigrade. Let's see the sources, uses and therapeutic of testosterone. It is produced by the men gonads, which is the major source for the production of the testosterone. Testosterone is soluble in ethanol, methanol, and it is used for primarily to treat symptoms of sexual dysfunction in men and women and hot flashes in women in additions with to increase the desire of libido. Let's see the last molecule which is presented on this page is DHT, that is dihydrotestosterone. The molecular formula of dihydrotestosterone is carbon 19 H30O2 and molar mass is 290.447 gram per mole. So there is a difference between testosterone and dihydrotestosterone, only the presence of double bond at carbon 4 positions, which is present in testosterone and however absent in the dihydrotestosterone, while all other structural features are the same as that of the testosterone. Most DHT is produced in the peripheral tissues like the skin and the liver, whereas most circulation dihydrotestosterone originates specifically from the liver. So these are the major sources from which dihydrotestosterone is generated. Dihydrotestosterone is soluble in dimethyl sulfur oxide and ethanol, and when we see its uses, DHT is biologically important for sexual differentiation of the male genitalia during embryogenesis, maturations of the sex organs, and scrotum at puberty, growth of facial, body and pubic hairs. And DHT is used for the development and maintenance of the prostate glands and seminal vesicles. Dear students, these are some important pharmaceutical brands of steroid molecules which we have missed in our previous lectures. So we discuss some important nutraceutical brands. The first brand is high lignin flaxseed complex. It is manufactured by Invite Nutraceuticals. We take 600 mg per day its dose and it's formulated in capsule dosage form. The activity of this brand is hormonal balance, cholesterol control and prevent hair loss. The second brand is Super Omega 3, which is manufactured by Life Extensions and we take 10 mg per day its dose. It is formulated in soft gel capsule. The activity of this brand is cholesterol control. The name of third brand is 7-HMR lignans. It is manufactured by Swenson Ultra and we take its dose 40 mg per day. It is formulated in capsule dosage form and the activity of this brand is electron enhancer. Let's see how different kinds of hormones are synthesized within the mitochondrial section, and we will discuss about the adrenal cortex as well as we will discuss about the enzyme which is responsible as a precursor for the synthesis of different kinds of hormones. The adrenal cortex is the outer region and also the largest part of the adrenal gland. It is divided into three separate zones: zona glomerulosa, zona fasciculata, and zona reticularis. Each zone is responsible for producing specific hormones. Now we will see how the cholesterol is changed into pregnenolone. Cholesterol is a hormone which is consisted of carbon 27 and by the effect of enzyme P450 that is a mitochondrial enzyme that catalyzes conversion of cholesterol to pregnenolone. This is the first reaction in the process of steroidogenesis in all mammalian tissues that specialize in the productions of various steroid hormones. So, in the second step, the pregnenolone is converted into 17-hydroxypregnenolone by the effect of enzyme 4P450 carbon 17, which is the single enzyme mediating both 17 alpha hydroxylase, steroid 17 alpha monooxygenase, and 1720 lyase activity in the synthesis of steroid hormones. It has been suggested that different P450 carbon 17 isozymase mediate these activities in the adrenal glands and testes. So, this enzyme converts the pregnenolone into 17-hydroxypregnenolone. In the next step, the 17-hydroxypregnenolone is changed into DHEA, that is dihydroepiandrosterone, by the effect of enzyme P450 carbon 17. DHEA, that is dihydroepiandrosterone, is a hormone produced by your body's adrenal glands. These are the glands just above your kidneys. DHEA supplements can be made from wild yam or soy. DHEA further rearranged into androsterone by the effect of enzyme 1720 lyase. Let's see how the pregnenolone and 17-hydroxypregnenolone is changed into progesterone and 17-hydroxyprogesterone respectively by the effect of enzyme 3-beta HSD150M. 3-beta hydroxysteroid dehydrogenase isomerase, that is abbreviated as 3-beta HSD, is an enzyme that catalyzes the biosynthesis of steroid progesterone from pregnenolone, 17-alpha hydroxyprogesterone, from 17-alpha hydroxypregnenolone, and androstenedione from dehydroandrosterone, that is DHEA, in the adrenal glands. It is the only enzyme in the adrenal pathway of corticosteroid synthesis that is not member of the cytochrome P450 family. It is also present in other steroid producing tissues including the ovary, testes and placenta. In humans, there are two 3-beta HSD isozymase encoded by the HSD3-beta1 and the HSD3-beta2 genes. 3-beta HSD is also known as delta 5,4 isomerase, which catalyzes the oxidative conversion of delta 5 3-beta hydroxysteroid to the delta 4 3-keto configurations and is therefore essential for the biosynthesis of all classes of hormone steroids, namely progesterone, glucocorticoids, mineralocorticoids, androgens, and estrogens. The 3-beta HSD complex is responsible for the conversion of pregnenolone to progesterone, 7-alpha hydroxypregnenolone to 7-alpha hydroxyprogesterone, DHEA2 androstenedione, androstenedione to testosterone, and androstenol to androstenedione. In this way, the pregnenolone is converted into progesterone by the effect of enzyme 3-beta HSD. Similarly, in the second step, the 17-alpha hydroxypregnenolone is changed into 17-hydroxyprogesterone by the effect of 17-20 hydroxylase enzymes. On this stage, the progesterone and 17-hydroxyprogesterone are changed into 11-DOC and 11-deoxycortisol by the effect of enzyme 21-hydroxylase. The enzyme 21-beta-hydroxylase is a cytochrome P450 enzyme that is involved in the biosynthesis of steroid hormones, aldosterone and cortisol. This synthesis takes place in the adrenal cortex, as you know. Specifically, 21-hydroxylase converts progesterones and sulfa 17-alpha hydroxyprogesterone into 11-deoxycorticosterones and 11-deoxycortisol respectively. So, the progesterone by the effect of enzyme 21-hydroxylase gives 11-DOC, that is called deoxycorticosterone, which is further converted into corticosterone by the effect of enzyme P450 aldosterone and P450 hydroxylase. In this way, the 11-deoxycorticosterol is changed into cortisol simply, and at the end, the corticosterone is changed into aldosterone by the effect of enzymes P450 11-beta aldo and 18-hydroxylase, as well as by the effect of enzyme 18-oxidase, which gives finally the hormones aldosterone. Let's see what is gonads. A gonad is a sex gland. A reproductive gland is a mixed gland that produces the gametes, that is sex cells, and the sex hormones of an organism. In the female of the species, the reproductive cells are the egg cells, and in the male, the reproductive cells are the sperm. The male gonad, the testicles, produces sperm in the form of spermatozoa. The female gonad, the ovary, produces egg cells. Both of these gametes are haploid cells. Let's see how different kinds of hormones are synthesized within the gonads. The cholesterol is a precursor which is used for the convergence of pregnenolone by the effect of 450 enzyme SCC. In the second step, the pregnenolone is further changed into 7-hydroxypregnenolone by the effect of enzyme 450 carbon 17, that is 17 hydroxylase, which have been discussed in the previous slide. The 7-hydroxypregnenolone is further converts into DHEA, that is dihydroepiandrosterone, by the effect of enzyme 1720 lyase. The DHEA is changed into androsterone by the effect of enzyme 1720 lyase. Let's see how pregnenolone and 17-hydroxypregnenolone is changed into progesterone and 17-hydroxyprogesterone respectively. The pregnenolone is changed into progesterone by the effect of enzyme 3-beta-HSD, and the 17-hydroxypregnenolone is changed into 17-hydroxyprogesterone by the effect of enzyme 1720 lyase. Let's see how androsterone is changed into testosterone and estrone. The androsterone is changed into testosterone by the effect of enzyme 17-beta-HSD and changed into estrone by the effect of enzyme 450 aromatase. The testosterone and estrone is further changed into DHT, that is dihydrotestosterone, by the effect of enzyme 5-direct, and estrone is changed into estradiol by the effect of enzyme P450 aromatase. So this is the general procedures for the biosynthesis of different kinds of hormones and the effect of different kinds of enzymes upon it. Let's see the functions of some steroidal hormones. Steroid hormones play an important role in carbohydrate regulations, which is carried out by the hormone glucocorticoids. It is also used in the mineral balance, in which mineralocorticoid is used for the said purpose. The hormones which is used for the reproductive functions is the gonadal steroids. Steroids also play an important role in the inflammatory responses. It is also used for the stress responses and bone metabolism. The hormones can also be used for the cardiovascular fitness, behavior, cognitions, and mood swings. Here in this slide, we will discuss the functions of some important hormones which have been derived from cholesterol. First, the androgens. It is strongest, that is testosterone, produced in the testes primarily, but weak androgens in adrenal cortex, that is anabolic steroid. It is used for the development of male secondary sex characteristics and prevents bone resorption. The second hormone is estrogen, which is produced in the ovaries primarily, but also in the adipose cells of male and females. It is used for the development of female secondary sex characteristics and also used to prevent bone resorption. The third hormone is vitamin D, not a steroid hormone, produced in the skin in response to UV light and processed to active form in kidney. It is used in the intestinal calcium absorption, promotes bone formation, and prevents phosphate loss by the kidneys. Let's see what are the carotenoids. Carotenoids are the organic phytoconstituents that are isolated from less to medium polar fraction of the plants, and these are yellow, orange, and red pigments which belongs to the category of tetraterpenoids. The tetraterpenoids consist of isoprenoid unit, and that unit is C5H8O. You can see over here some important examples, and that examples are lycopene, canthaxanthine, astaxanthin, and beta-carotene. The first molecule, that is lycopene. Lycopene is a bright red carotenoid hydrocarbons found in tomatoes and other red fruit and vegetables, such as red carrots and watermelons, grapefruits and papayas, but it is not present in strawberries and cherries. Although lycopene is chemically a carotene, however, it has no vitamin A activity. When we see its uses, the lycopene is used as a food supplement. However, it is used in high blood pressure, high cholesterol, cancer, and many other conditions, but there is no good scientific evidence to support these uses. The second molecule is canthaxanthine. Canthaxanthine was first extracted from an edible mushroom in 1950s. It has since been identified in sea trout, algae, daphnia, salmon, brine shrimp, and several species of flamingo. In addition to these natural sources, canthaxanthines can be synthetically produced from acetone. When we see its uses, people use it as medicine. Canthaxanthin is used to reduce sensitivity to sunlight, that is called photosensitivity, experienced by people who have a great genetic disease called erythropoietic protoporphyria. In these people, sunlight can cause skin reactions such as itches and rashes. The next molecule is astaxanthin. It is found in salmon, lobster, crab, and crayfish, as well as in krill oil and algae. When we see its uses, astaxanthin is taken by mouth for treating Alzheimer's diseases, Parkinson's diseases, stroke, high cholesterol, liver diseases, age-related macular degenerations, age-related vision loss, and preventing cancer. The next molecule is beta-carotene. Beta-carotene is a red-orange pigment found in plants and fruits, especially carrots and colorful vegetables. When we see its uses, the human body converts beta-carotene into vitamin A, that is called retinol. Beta-carotene is a precursor of vitamin A. We need vitamin A for healthy skin and mucous membranes, our immune system, and good eye health and vision. Vitamin A can be sourced from the food we eat. Carotenoids are soluble in organic solvents and insoluble in water. Now we see the identification test of the carotenoids. Here we perform two tests. In the first test, the sample is taken in chloroform and then added concentrated sulfuric acid along the side walls of the test tube. So we get the blue color. It means carotenoids are confirmed. In the second test, the sample is taken in chloroform and added antimony trichloride, which gives the blue color, which indicates the presence of carotenoids. Common sources of carotenoids are pumpkin, carrots, spinach, corn, tomatoes, bacteria, and fungi. Some phytoconstituents have biological activities that are antioxidant, anti-cancer, and anti-inflammatory. The phytoconstituents that are present are alpha-carotenes, beta-carotenes, and lycopene, lutein. Carotenoids play an important role in biosynthesis, imparts color to plants, a rich source of vitamin A used in various herbal products. Now we discuss some important pharmaceutical brands in which carotenoids are used as food supplement. So the first brand is I Factor Formula, that is manufactured by Natural Factors, and we take its dose 262 mg per day. It is formulated in capsule dosage form, serving per container 90 capsule. The activity of this brand is protective visions as an anti-inflammatory. The second brand is Beta Care All. It is manufactured by Natural Factors and we take its dose 25 mg per day. It is formulated in soft gel capsule. The activity of this brand is support immune system and cardiovascular systems for healthy skin, bone, teeth, and vision. The last brand is Astaxant in Plus. It is also manufactured by Natural Factors and we take its dose 5 mg per day. It is formulated in soft gel capsules and its activity is antioxidant. Let's see the isolation and purifications of steroids. This is the specific method for the extraction and isolation of steroid molecules. In this method, we will take hot boiled egg yolk in which 1 gram of potassium carbonate is added. After that, 5 gram of sand and 10 ml of methanol is added. Whole material is grinded and we will get the soft scrambled egg material. In this material, we will add 20 ml of cyclohexane and stir it for some time. After that, we will take the cornmeal mush material. The whole material is then formed for reflex and evaporation. After evaporations, we will get the evaporated egg mixtures in which we will add 30 ml of methylene chloride. Now, this material is subjected for column chromatography in which we will take 15 gram of flash silica gel and layer of sand on the top. So, the column is packed. After that, the whole column is eluted with 30 ml of methylene chloride. Now, it is rinsed with ethanol with the help of petroleum ether. After that, again, the elution is carried out with the help of ethanol and petroleum ether with the specific ratio. So, in this way, from the column chromatography, we will get total 15 fractions and each fractions have 10 ml of volumes. Now, each fraction is subjected for thin layer chromatography for the identifications of the pure compound as well as for the presence of steroid molecules. So, the cholesterol will be spread into 6 to 16 fractions. Now, after the evaporation of cholesterol fractions, we will get the cholesterol crystals in the round bottom flask. Now, the steroid, which is in the form of cholesterol, is recorded for weight and then taken its melting point. After that, the cholesterol is identified with the help of specific test for the conformations of the steroid molecules. So, this is the specific method for the isolations and purifications and identifications of the steroids natural products. Dear students, these are the assignments and the quiz which you would have to submit at your earliest. That is, describe the function of hormones. Also share its some nutraceutical brands with key ingredients. The second assignment is discuss the mode of mechanism of cholesterol in human physiology. Thank you.